Hello dear students, welcome back to BioVidyalaya. I hope you all are doing very well. This is part 2 of our CSIR NET PYQ series. Here I am coming with some questions based on our previous lecture video, that means cell adhesion molecule. I think you all watched our cell adhesion molecules lecture video. If you didn't watch, then the link is here in the below description. Please go through that. So let's begin. This is the first question from CSIR NET December 2017. Question is, several types of molecules including the transmembrane glycoproteins can function as matrix receptors and co-receptors. However, the principal receptors on animal cells for the binding most extracellular matrix proteins are the integrins. Which of the following statement is not true for integrins? Statement 1. Integrins are transmembrane linker protein that link to the cytoskeleton. Statement 2. An integrin molecule is composed of two non-covalently associated glycoprotein subunits alpha and beta. Both subunits span the cell membrane with short intracellular C-terminal tails and large N-terminal extracellular domains. Statement 3. The extracellular portion of the integrin dimers bind to specific carbohydrate residues in extracellular matrix proteins or to ligands on the surface of other cells. Statement 4. The intracellular portion binds to a complex of protein that form a linkage to the cytoskeleton. This question is based on integrins. We already studied that in our previous lecture video. If you didn't watch that video, then the link is here in the below description. Please do watch. So here some statements about integrins are given. From these statements, we have to select which statement is not true. Which of the following statement is not true for integrins? Okay. So first statement, integrins are transmembrane linker protein that link to the cytoskeleton. Is it correct? Yes, this is correct. We studied that. Cytoplasmic portion. This is cytosolic domain. That means cytoplasmic portion. Cytoplasmic portion of integrin molecule bind to adapter protein and then it will interact with actin filaments inside the cell. Actin filaments are part of cytoskeleton. Okay. So this is correct. Second statement. An integrin molecule is composed of two non-covalently associated glycoprotein subunit alpha and beta. Both subunits span the cell membrane with a short intracellular C-terminal tail and large N-terminal extracellular domains. See here, this is the structure of integrin. We know that integrin is composed of two subunits alpha and beta. That's why it is a heterodimer. Integrin is a heterodimer. This is the extracellular portion and this is the cytosolic portion. Okay, that means C-terminal tails. C-terminal tail. So this statement is also correct. Next statement 3. The extracellular portion of integrin dimers bind to specific carbohydrate residues in extracellular matrix proteins or to ligands on the surface of other cells. This is not correct because we studied that integrins can bind to variety of molecule like fibronectin, fibrinogen, collagen and glycoprotein. Okay, so this statement is not correct. Next is the intracellular portion bind to complex of protein that form linkage to the cytoskeleton. Yes, this is also correct because cytoplasmic portion, that means C-terminal N, can bind to adapter proteins like vinculin, alpha, actinin, taurine, etc. and that will bind with actin filament. That means cytoskeleton. So this statement is also correct. So which statement is not true? Yes, this is statement 3. So the answer is option 3. Okay. Match the following questions are very easy to solve even if you are able to match only 2 of them. Then also you can easily solve such type of question. So let's see how to solve this question. Here in column A they given cell adhesion proteins names, and in column B they given functional characteristics. So first we can read column B and we can match according to that. So here in I they given lectins that mediate a variety of transient cell-cell adhesion interaction in the bloodstream. As we studied that selectins have conserved lectin domain that are binding to oligosaccharide on another cell. They are playing main role in bloodstream. So here it is selectin. Where is selectin? D. So here it is D. I will write here D is I. Okay. Next, contains extracellular Ig-like domain and are mainly involved in the fine tuning of cell-cell adhesive interactions during development and regeneration. Yes, this is immunoglobulin superfamily. It is very easy to answer because here it is given Ig-like domain. So it is very easy to answer this. This is C. So C is 2. So let's see the options. Here in option, where is C2? Here it is 3, it is 4, and here it is 1, and C. Here C2 is there. Next is D1. Here also D1 is there, but here C is 4, isn't it? So here it is D1. Here C2 is there and D1 is also there. So correct answer is option 4. Okay. Now we only matched 2 and now we got the answer also. So it is very easy to answer. Match the following questions. Here we can see the third one. Mediate calcium dependent strong homophilic cell adhesion. Which one is calcium dependent? Cadherin. Selectin is also calcium dependent, but we already matched that with 1. And next is 4. Transmembrane cell adhesion proteins that act as extracellular matrix receptors. Now only we discussed that integrins are principal receptor used by animal cells to bind to the extracellular matrix. Okay. So this is A. So A is 4, B 3, C 2 and D 1. So correct answer is option 4. This is the question from December 2014. Which one of the following statements about cell-cell interaction is not true? Statement A. Cadherins are transmembrane linker proteins which carry out calcium-mediated adhesion between adjacent cells. Statement B. Integrins are transmembrane adhesion proteins that mediate homophilic adhesion through actin and intermediate filaments. Statement C. Selectins are cell surface lectins that mediate a variety of transient cell-cell adhesion interactions in the bloodstream. And statement D. ICAMs, intracellular cell adhesion molecule, and VCAM, vascular cell adhesion molecules, are members of immunoglobulin superfamily. Option 1 only A, option 2 only B, option 3 both C and D, option 4 both A and D. So from this following statement, we have to find which statement is not true. Okay, which statement is not true. So first statement, cadherins are transmembrane linker protein which carry out calcium mediated adhesion between adjacent cell. Yes, we studied that catharins are involved in calcium-dependent cell-cell adhesion. Okay, so this statement is true. Next statement B. Integrins are transmembrane adhesion protein that mediate homophilic adhesion through actin and intermediate filament. Integrins can interact with actin and intermediate filament, but they are involved in heterophilic interaction, not in homophilic interaction. So this statement is not true. Next statement C. Selectins are cell surface lectin that mediate a variety of transient cell-cell adhesion interaction in the bloodstream. Yes, this is also correct because we studied that selectins are present in endothelial cells. Okay, that can interact with leukocytes. So this statement is correct. Next statement D. ICAMs and VCAMs are members of immunoglobulin superfamily. This is also true. Which are immunoglobulin superfamily CAMs? ICAM, VCAM and NCAM. Okay. So this statement D is also correct. So here we have to find which statement is not true. Which is not true? Yes, statement B is not true. So correct answer is only B. So answer is option 2, only B. Okay. This is one part B question from CSIR NET December 2012. Which of the following is an intracellular anchor protein? Options are option A vitronectin, option B vinculin, option C integrin, option D elastin. So here we have to find which is an intracellular anchor protein. Anchor protein means adapter protein, that is linking cytoskeleton with CAM. Okay. So here vitronectin. Vitronectin is extracellular matrix protein. Okay, extracellular matrix protein. Integrin is CAM, cell adhesion molecule. And elastin is also extracellular matrix protein in connective tissue. And vinculin is intracellular anchor protein, that is adapter protein, that is anchoring or that is connecting actin with CAM. Okay, so this is correct answer. So option B, vinculin is the correct answer. This is very easy question. This is from CSIR NET June 2011. Question is, which of the following molecules is involved in calcium dependent cell-cell adhesion? Options are option A calmodulin, option B catharin, option C NCAM and option D calpain. So among these four options, which are cell-cell adhesion molecule, CAM molecules? Yes, catharin and NCAMs are CAM molecule. Among these, which are calcium dependent? Yes, catharin is calcium dependent. Okay, and this NCAM is calcium independent. Okay, this is coming under, NCAM is coming under immunoglobulin like superfamily and they are calcium independent. So our answer is option B, catharin. Okay. And here calmodulin. Calmodulin is involved in cell signaling and that is also calcium dependent, and calpain is involved in apoptosis and that is also calcium dependent. Okay, that need calcium for their activation. So here our answer is option B, catharin. Catharin is calcium dependent cell-cell adhesion molecule. Okay. This is also one match the following question and this is from CSIR NET December 2018. So here first we can read column B and we can match according to that. Okay. The chief endothelial cell protein that are recognized by white blood cell integrins and member of immunoglobulin superfamily. So let's see which is immunoglobulin superfamily. See, ICAM. ICAM is the immunoglobulin superfamily. So this is C. C is I. Okay. See here, Ci is there. Okay. Next is cell surface carbohydrate binding protein that mediate a variety of transient cell-cell adhesion interaction in the bloodstream. Which is correct? Carbohydrate moiety is present in selectin, isn't it? So this is D. So D is 2. Already we got the answer C1D2. This is option C. Okay. So let's see other statements also. 4-pass transmembrane protein, which is the major constant of gap junction forming a continuous aqueous channel. Okay. Here we can see that this is connexin. Connexin is gap junction protein. Okay. So this is A3. Okay. And next is plasmodesmata. It is the only class of intracellular junction in plant cell that directly connect the cytoplasm or adjacent cell. Plasmodesmata is the cell junction present in plants. So B is 4. So which is the correct answer? Option C. Okay. If you like this video and find it helpful, then please click the like button, share this video with your friends, and also don't forget to subscribe this channel for getting more videos like this. Keep learning. Thank you.